Every second, millions of cells in your body are dividing, and before a cell can divide, it must perform one of the most essential tasks of life, replicating its DNA. Imagine copying an encyclopedia with 3 billion letters without making mistakes in just a few hours. That's what happens inside your cell every single day. In this video, we are going on a molecular journey. We will zoom into the cell nucleus, unravel the DNA helix, meet the enzymes that act like molecular machines, and see step by step how life's blueprint is duplicated. By the end, you will understand not only how replication works, but also why it's so central to health, disease, and evolution. So let's talk about a little bit about DNA. DNA, or the deoxyribonucleic acid, is the genetic instruction book of life. Every cell in your body except red blood cells carry the same DNA, tightly packed into the nucleus. DNA is a double helix, two-stranded, twisted around each other, made up of 4 bases: A, T, G, C. The sequence of these letters encode all the information to build and maintain life. But when a cell divides, each daughter cell needs an identical copy for the information. That's why DNA replication is so crucial. It ensures that genetic instructions are faithfully passed on. So, replication begins at the special regions called origin of replication. In bacteria, there is just one origin, but in human cells or in the eukaryotes with billions of base pairs, there are thousands of origins working at the same time. Proteins called initiators recognize these regions, unwind a small portion of DNA, and form structure called the replication bubbles. From these bubbles, replication fork spread outward in both directions. This massively sped up the process. Otherwise, replication would take weeks instead of hours. Next comes the molecular motor, or the helicase. This enzyme acts like a zipper slider, racing along the DNA and unwinding the 2 strands. It breaks the weak hydrogen bonds between bases, exposing single strand that can now be copied. But as helicase works, the DNA ahead of it twists tighter and tighter, creating tension, just like twisting a rubber band. To relieve this stress, another enzyme called topoisomerase makes temporary cuts in the DNA, untangles it, and then reseals it. In the next step, we can say that once the DNA strands are separated, single DNA strands are unstable. They want to snap back together. That's where single strand binding proteins, or SSB proteins, step in. They coat the exposed strands, preventing repairing and protecting DNA from damage until replication is complete. In the 4th step, the twist comes. The main copying enzyme, DNA polymerase, to be specific DNA polymerase 3, cannot start from the scratch. It needs a small piece of RNA as a starting block called the primer. The primer is made by the enzyme primase, another enzyme that synthesizes shorter RNA sequence complementary to the DNA template. Once primers are in the place, DNA polymerase 3 can jump into the action. Now, the star of the show arrives, DNA polymerase 3. This enzyme adds new nucleotides, one by one, to the exposed template strand. But here's the catch. DNA polymerase can work only in the direction of 5' to 3'. Because of this, the 2 strands are copied differently. The leading strand is built smoothly, continuously, towards the replication fork. But the lagging strand, however, is built in small fragments called the Okazaki fragments, moving away from the fork. These fragments are later stitched together. Once the Okazaki fragments are laid down, gaps remain between them. DNA ligase acts like a molecular glue, sealing these fragments into one continuous strand. The final step is the proofreading or the error correction. Replication must be nearly flawless. DNA polymerase has a built-in proofreading function. If it adds the wrong base, it can backtrack, remove the error and replace it with the correct one. This keeps the error rate extremely low, about one mistake in 10 million bases, and even if errors slip through, other repair system can catch them later. Here you can see the example. T, that means thymine, mistakenly bind with guanine or G. But we know that thymine binds with adenine. So the DNA polymerase, to be specific DNA polymerase 1, will remove the guanine and it will insert adenine with the thymine. In the next step, we can say that at the end, the cell has two identical DNA molecules. Each new DNA is made up of one original strand and one newly synthesized strand. That is called semi-conservative replication. This method ensures both accuracy and efficiency. It's like making a photocopy where half of the original page is still part of the new one. Here you can see that the dark blue color is the main DNA and the light blue color strands are newly formed, and that's why it is called the semi-conservative method. Let's look at the summary in one picture. In this picture, you can see that there is a replication fork from where the replication will take place. Helicase helps in unwinding the two strands, and the SSB proteins will stabilize the single strand, but the SSB proteins are not shown in this picture. Again, the primer will insert by the primase, and DNA polymerase helps in the replication process. For your easy understanding, I have inserted one video. Just have a look at this. Here you can see that in the origin of replication, first the helicase enzyme has bind and it is unwinding the two strand, and single strand binding proteins are stabilizing this. Primase has come and it is inserting the RNA primer. Now DNA polymerase will come and it will start binding the other nucleotide in the 5' to 3' direction, and that strand will be called the leading strand, and the opposite strand, that means the lagging strand, will make the Okazaki fragments. Here you can see the details. Here you can see again the primase came and again the DNA polymerase 3 makes the lagging strand or the Okazaki fragment, and DNA polymerase 1 will come to see any error is there, and the ligase will finally binds and seal the gap. Let's have a look how the bases bind one by one by the DNA polymerase. Here you can see that the adenine base is open and it will bind with thymine. So the nucleotide will come, it will make the hydrogen bond, 2-phosphate will be removed and it will make the bond. Without accurate DNA replication, life couldn't exist. Cells would lose information, mutations would pile up, and organism could grow or reproduce. DNA replication also connect to cancer. Uncontrolled cell division can fueled by replication errors. Aging, as the cell replicates, DNA ends shorten, leading to the telomere erosion. In biotechnology (PCR), the method used in labs to copy DNA is based on the same principle that of the replication. And in evolution, rare replication errors or mutations create genetic variation, the raw material of evolution. So, what happens inside a cell during replication? It's a perfectly choreographed dance of enzyme where helicase unzips DNA, primase lays the foundation, polymerase builds new strands, ligase seal the gap, and proofreading ensures accuracy. This microscopic process is repeated billions of times in your body every single day, a constant act of molecular engineering that keeps life going. Next time you think about your cells dividing, remember inside you right now millions of tiny copy machines are at work, faithfully duplicating life's blueprint. Hope you like this video. Thank you for watching.